Hi. Today we're going to talk about gene expression in bacteria. This is a very fundamental process, as most of the processes are that we discuss in genetics. But this one in particular is interesting because what we have learned about gene expression in bacteria has influenced our understanding of how genes are regulated in all organisms. So understanding these concepts today will help you understand how genes are regulated in nearly any system that we study, or you decide to study in the future, perhaps. So I hope you enjoy the lecture. Please think of questions for question time. And here comes the voiceover. Gene regulation is critical for the function of all cells. Bacteria are no exception. Here is Bonnie Bassler, an HHMI investigator who has uncovered mechanisms of how bacteria communicate with each other. We will discuss her important work in the second half of the lecture. Gene regulation is essential. The control of gene expression is critical for both unicellular and multicellular organisms. In unicellular organisms, gene regulation is necessary to turn genes on and off in response to environmental changes. In multicellular organisms, gene regulation specifies cell differentiation and function. Gene expression can be controlled at multiple levels, from the DNA to the final functional protein. For example, the state of the DNA, how compact it is, can control the level of gene expression. So if you alter the structure and relax the DNA coiling, you can get increased transcription. Once a gene is transcribed, it is processed and spliced. This processed mRNA has a certain half-life, which is related to its, of course, stability, and must be translated. The translated protein is initially inactive until it properly folds, and some proteins actually are modified post-translationally, and this can change the activity of the protein. So at any of these steps, you can regulate the function of a gene. DNA binding proteins contain specific domains that can interact with DNA, sometimes in a sequence-specific manner. These domains are contained in proteins that regulate the expression of genes. For example, the Helix-turn-helix motif, which are contained by many bacterial regulatory proteins, although similar motifs also exist in eukaryotic proteins. They consist of two alpha helices, and they bind to the major groove of the DNA double helix. You have zinc finger motifs in DNA binding proteins. These are found in eukaryotes. And there is a loop of amino acids with zinc at the base. And these motifs bind to the major groove of DNA. Another example is leucine zipper motifs. This is a motif found in eukaryotic transcription factors, and it's a helix of leucine residues and a basic arm. These motifs bind to adjacent major grooves of DNA. Now, these motifs help proteins regulate gene expression. And in bacteria, we have a really great understanding of gene expression, in part because of the discovery of the Operon. And the Operon is regulated by a protein, a regulator protein. And this operon was described by Jacob and Monod in 1961, and they later won the Nobel Prize for their work. So let me explain a little bit about what an operon is and how this particular operon works. An operon is a group of structural genes plus sequences that control transcription. For example, the regulator of the lac operon is transcribed from a separate region of the bacterial chromosome. So this gene has its own promoter and it encodes a regulatory protein. This regulator protein binds to the promoter operator region of the lac operon, and when it's not around, you can get transcription of the genes in the operon, and they're transcribed as one mRNA altogether. The regulator protein may bind to the operator site to regulate the transcription of the mRNA. And in this case, it would repress it. And the products of the mRNA catalyze reactions in a biochemical pathway. In the case of the lac operon, this is about the metabolism of lactose. So here we have the lac operon. In the absence of lactose, the active regulator protein, the repressor, is transcribed and translated. In the absence of lactose, the regulator protein, which is a repressor, binds to the operator and inhibits transcription. So you get no transcription. In the presence of lactose, you still get transcription and translation of the active regulator protein. But when lactose is present, because of beta-galactosidase, some of the lactose is converted to allolactose, which then binds to the regulator protein, making the protein inactive. Therefore, the regulator protein cannot bind to the operator, and the structural genes are transcribed and translated. And this allows lactose to be made into glucose and galactose. Now here's an animation of the whole process of the regulation of the lacto-operon.
To utilize lactose as an energy source, E. coli must produce the enzymes necessary for its metabolism. The genes for these enzymes are adjacent to one another on the bacterial chromosome and are transcribed together. A group of bacterial genes that are transcribed together is called an operon. At one end of the lac operon is a set of structural genes, Lac-Z, Lac-Y, and Lac-A, that encode the enzymes necessary for metabolizing lactose. These three genes have a common promoter. The regulator gene, Lac-I, is located upstream of the structural genes. The Lac-I gene encodes a repressor protein and is controlled by its own promoter. This repressor protein may bind to a region of the operon called the operator. The operator overlaps the 3' end of the promoter and the 5' end of the Lac-Z gene. The lac operon is inducible, that is, the structural genes are expressed only in the presence of lactose. Active repressor proteins are encoded by the regulator gene. In the absence of lactose, these repressors bind to the operator and prevent RNA polymerase from initiating transcription. When lactose is present, it is readily converted into allolactose, which may then come in contact with the repressor protein. Allolactose binds to the repressor protein and causes a conformational change that prevents the repressor from binding to the operator region of the DNA. With the operator site open, RNA polymerase is able to bind to the operon promoter and transcribe the structural genes. The resulting mRNA is then translated, leading to the production of the 3 enzymes, beta-galactosidase, permease, and transacetylase, encoded by Lac-Z, Lac-Y, and Lac-A, respectively. Lactose is a carbohydrate found in milk. It can be metabolized by E. coli bacteria that reside in the mammalian gut. Because lactose is too large to diffuse across the bacterial membrane, it must be actively transported into the cell by permease, encoded by the LAC-Y gene of the lac operon. To utilize lactose as an energy source, E. coli bacteria use the enzyme beta-galactosidase, encoded by the LAC-Z gene of the lac operon, to break lactose down into glucose and galactose. The function in lactose metabolism of the third enzyme, transacetylase, also produced by the lac operon, is not yet known.
The plasmid contains the promoter and regulator gene, as well as the promoter and the operator, and the lac operon. So let's change what the plasmid contains. What if the plasmid has no regulator gene, a non-functional version of the regulator gene, and it has a normal version of the promoter and operator region? Let's view.
The LAC-I plus gene produces active repressor proteins. The LAC-I minus gene produces mutant repressor proteins. The mutant repressor proteins are unable to bind to the operator regions of either operon, even in the absence of lactose. However, the active repressors are able to bind to operator regions of either operon where they prohibit the binding of RNA polymerase. Because RNA polymerase cannot bind, the structural genes are not expressed. In the presence of lactose, and thus allolactose molecules bind to the active repressor proteins. As a result of this allosteric inhibition, the repressor proteins can no longer bind to the operator regions. When the operator regions are open, RNA polymerase can bind and transcribe the structural genes. The transcribed mRNAs are translated into the enzymes needed for lactose metabolism.
So let's try another scenario. Let's say that we have a normal LAC-I in the plasmid, and we have a constitutive promoter and operator mutation in the plasmid. Let's see what we get.
The LAC-I plus genes produce active wild-type repressors. The repressors are transacting and may attempt to bind to either operator. In this case, a repressor binds to the wild-type operator, but is unable to bind to the mutant operator. Repressor bound to the lower operon blocks RNA polymerase from binding. However, no repressor is bound to the mutant operator to impede the polymerase. Thus, RNA polymerase transcribes the structural genes of the upper operon, and the mRNAs are then translated into the lactose metabolizing enzymes, even in the absence of lactose. In the presence of lactose, and thus allolactose molecules bind to the active repressor proteins. As a result of this allosteric inhibition, the repressor proteins can no longer bind to the wild-type operator. Now both operons are available for transcription. RNA polymerase transcribes the structural genes. The mRNAs are then translated into the enzymes necessary for metabolizing lactose.
So let's say that we have a wild-type version of the operator region, and we have now a special version of the LAC-I gene. This is a special type of mutation, and you'll see how.
The LAC-I plus gene produces wild-type repressor proteins. The LAC-IS gene produces mutant repressors that have no allolactose binding site. These repressors are called superrepressors. The superrepressors are transacting and can bind to either operator region. The wild-type repressors are also transacting and are just as likely as the superrepressors to bind to either operator. With the repressor proteins bound to the operator regions, RNA polymerase is unable to bind and the structural genes are not expressed. In the presence of lactose and thus allolactose molecules bind to the wild-type repressors. The allosterically inhibited active repressors can no longer bind to the operator regions. Allolactose cannot bind to the superrepressors, so these repressors are still available to bind to the operators of both operons. With the superrepressors bound, RNA polymerase cannot initiate the transcription of the operons. The structural genes are not expressed, even in the presence of lactose.
Let's see if we can overcome the effect of the superrepressor. So we have the superrepressor, and then we have the constitutive promoter operator region in the plasmid.
The LAC-I plus gene produces wild-type repressor proteins, whereas the LAC-IS gene produces mutant repressors that have no allolactose binding site. These repressors are called superrepressors. Both repressor types are transacting and could potentially bind to either operator region. However, neither the mutant superrepressors nor the wild-type repressors are able to bind to the mutant operator. Repressor bound to the lower operon blocks RNA polymerase from binding. However, no repressor is bound to the mutant operator to impede the polymerase. Thus, RNA polymerase transcribes the structural genes of the upper operon, and the mRNAs are then translated into the lactose metabolizing enzymes, even in the absence of lactose. In the presence of lactose and thus allolactose molecules are unable to bind to the superrepressors. Allolactose can allosterically inhibit the wild-type repressors so that they can no longer bind to the wild-type operator. The superrepressors are still available to bind to the wild-type operator and inhibit transcription. Because the superrepressor cannot bind to the mutant operator, RNA polymerase is free to transcribe the upper operon. The resulting mRNAs are then translated into the enzymes necessary for metabolizing lactose.
The tryptophan operon is regulated by attenuation. So this is a different mechanism for controlling gene expression. So when tryptophan levels are low, the RNase begins transcribing the DNA and produces region 1 of the 5' UTR of this operon. A ribosome attaches to the 5' end of the 5' UTR and translates region 1 while region 2 is being transcribed. The ribosome stalls at the trip codons in region 1 because tryptophan is low. Because the ribosome is stalled, region 2 is not covered by the ribosome, and region 3 is transcribed. When region 3 is transcribed, it pairs with region 2. When region 4 is transcribed, it cannot pair with region 3 because region 3 is already paired with region 2. You can see in the structure this hairpin that is formed. So the attenuator never forms and transcription continues. When tryptophan levels are high, the RNA polymerase begins transcribing DNA, producing region 1 of the 5' UTR as before. So the ribosome binds to the 5' end of the 5' UTR and translates region 1, while region 2 is being transcribed. RNA polymerase transcribe region 3. The ribosome does not stall at the trip codons because there's plenty of tryptophan around. And then the ribosome covers part of region 2, preventing it from pairing with region 3. Region 4 is transcribed and pairs with region 3, producing the attenuator that terminates transcription. So this loop prevents transcription from happening. So now I'm going to present you with an animation of how the trip operon works, and I think this will be a little clearer for you to see.
The trip operon in E. coli contains 5 structural genes corresponding to enzymes that convert choresmate into tryptophan. The TRPE gene contains a 5' untranslated region, or UTR, that plays an important role in the regulatory mechanism called attenuation. The 5' UTR contains 4 regions. Region 1 encodes a short peptide containing tryptophan amino acids. When tryptophan levels are high, region 3 pairs with region 4. This structure terminates or attenuates transcription. When tryptophan levels are low, region 2 pairs with region 3. This structure does not terminate transcription. Let's examine how these alternate structures arise. RNA polymerase begins transcribing the DNA. Closely following RNA polymerase, a ribosome begins to translate region 1. When tryptophan is abundant, the ribosome doesn't slow down at the tryptophan codons. By the time region 3 is transcribed, the ribosome has already moved past the tryptophan codons to partly cover region 2. Because region 3 is prevented from pairing with region 2, it pairs with region 4 instead to produce the attenuator, which terminates transcription. The structural genes are not transcribed, and therefore no additional tryptophan is synthesized. When tryptophan is scarce, the ribosome stalls at the tryptophan codons. Therefore, region 2 is not covered when region 3 is transcribed, allowing regions 2 and 3 to form a hairpin that does not terminate transcription. Because region 3 is paired with region 2, it cannot pair with region 4 and form an attenuator. Transcription continues. RNA polymerase transcribes the structural genes, which are translated into enzymes, which in turn synthesize more tryptophan. You can mimic the effect of tryptophan levels by determining how long the ribosome pauses at the trip codons. Click on the ribosome when indicated and hold down the mouse button for as long as you would like the ribosome to pause at the trip codons. Can you cause attenuation and anti-termination?
Okay, let me give this a try. I'm holding it down. And the structural genes are being translated. Now just quickly, all right, now let go. Let me do this really quickly. So now that you've heard about the TRP and the LAC operons, I think it's a good time to take a break. Here's a painting by Elizabeth Peyton. Relax, go for a walk, take a little break, and then come back, and then we're going to talk about Bonnie Bassler and her work with quorum sensing and the Lux operon. I think Bonnie Bassler is a really interesting person, and I find her story inspirational. So I'm going to share it with you. I hope you enjoy it.
I don't come from a scientific family. I'm the only scientist in my family. And I grew up as a regular kid. I have a brother and sister, and I'd stay-at-home mom. My mom was, she was interesting because she was one of the first people in her family to ever go to college. And at that time, when my mom was in college, which was in the 50s and early 60s, a woman could be either a teacher or a nurse, and those were the only two choices. And so she became an English teacher, but I do recall her telling me, but you can be anything you want, Bonnie, so just pick whatever you like. And I think she really inspired me to not be, afraid if I like science just because we didn't have any background in science or, nobody in my family knew science. She just said, if you like it, try it. I was a regular kid in high school. I was a huge athlete. I have had 18 varsity letters, I'm proud to say, none of which I could get today. I did sports, I was in Girl Scouts, I worked in the mall at a clothing shop to make money, I babysat. It was just kind of a regular life. It was not a rarefied life with academics. And I went to UC Davis to college because it's a very famous veterinarian school. As part of the bio major, you had to take biochemistry. So in my sophomore year, I signed up for this Biochem 101 class. And in that class, for the first time, it became this business of doing puzzles, where instead of memorizing, like the circulatory system, they would give you these problems about how cells work or how energy got generated or how molecules got made. And so I went to my advisor and I changed my major to biochemistry. And I started taking biochemistry and chemistry and molecular biology and all of the classes that went along with that major. And just from that moment on, I was on this path that I never looked back.
TV so far, so I hope you guys are all set up.
I have to check
My current family consists of 13 20 to 30-year-olds. That's like my big extended family. I also have a real family. I have a husband. He's an actor in New York City, so he's an artist. And he gets what I do as my craft, like the art of science, right? And I think this is why we like each other. He gets that I have that exact same feeling about my work. We both have a really nice life because he gets to be a voyeur into the life of the scientist, and I get to have a much more artistic life than I would have on my own. And so that's actually really nice in terms of spare time, right? Sort of putting a foot in the other person's world. When I was younger, and I would see all these famous scientists, and I would think, gosh, they're so smart and they know so much, right? I will never be like that. And now people look at me like that, like, oh, look at Bonnie. She's like a big shot scientist. I mean, I hope they do. She's a big shot scientist. She's really smart. She's made these contributions. She's made accomplishments, but I'm still the same person. And what I realize now is all of those people are just older, right? So that's me now. Like, you get better at it. It's like any job. Like, I have been around a long time now, and I've had time to learn things and amass this knowledge that I didn't have 20 years ago. And so when young people come to my lab, all I have over them is some common sense about, how these things work and about how just over time you get better at it. I still have my heroes. And so whatever these scientists are your heroes, it's not that they're intrinsically smarter. They're just better trained because they've been at it longer, right? I am terrified of the young people in my lab because I see them getting what I got, but moving past me, and it's great. That's the future. And so I think this idea that I can never be like so-and-so, whoever your scientist hero is, you are like that. You know, you're just more raw. You know, I still can't believe I'm a professor. You know, I still can't believe this is my job, right? But I haven't been found out yet. Like, it's going okay. And so I'm just gonna sort of keep going through and try my hardest and see where it goes.
Bonnie Bassler has done a lot of work on quorum sensing in bacteria. We're going to take a look at a section of her lecture on her work.
Already alluded to, they're everywhere on Earth. And so what scientists can do is go to the bottom of the ocean where the magma is roiling up from the center of the Earth like a volcano. It's 300 degrees. All of this metal smoke is coming out of these vents. And what do we find there? Bacteria living and thriving. Likewise, we can go to the coldest places on the Earth. So scientists have looked under layers of ice that have not thawed in tens of thousands of years, and we find microbes. So these are environments that we couldn't exist in, and yet these bacteria manage to occupy those niches. And so probably you and I don't get to go to those places very much, but we do get to be around here. So now I want to move into what these bacteria mean to you. And so this is just a picture of a person's arm. And what we've done is to take a micrograph of the elbow. So look under a microscope at the skin on a person, random human being's elbow. And what I hope you can see is that there are dozens of species of bacteria there. So this is how you are as well. And so each of those different shapes and color is a different species of bacteria. So on your skin, just in this one little patch, you have dozens of bacterial species. A few more on your teeth. So you guys, I hope, brush your teeth every morning, right? You clean your teeth because you have those little sweaters on them. Those sweaters are called biofilms. They are made up of bacteria. There are 600 species of bacteria on your teeth every morning. And so the final picture shows what your teeth look like before you brush them. And these bacteria are not willy-nilly. They are in an architected community where one guy sits next to the correct neighbor, and so on and so on. They are an amazing community that is working together and flourishing. The middle picture is to inspire you to change your toothbrush kind of frequently. That's just the bristle of a toothbrush because you're brushing these bacteria off. So you have bacteria all over you, all of your life. It's this invisible world that covers your body, and they are your body armor that keeps you safe from environmental insults. So they cover you and they protect you from bad and harmful things in the environment. And so so you are covered with them, but I want to tell you about relative numbers of you versus bacteria. So what we've done here, this is my idea of a human being and the cells that make up a human being. So we know how many cells are in a human being. There's about 1 trillion cells that we each need to make us alive and do all the things that we do. And so what we've done is to draw those little circles. Each of those circles represents a fraction of the human cells in a typical body making a live person. So 1 trillion human cells. Well, it turns out at any time of your life, you have 10 times more bacterial cells than human cells. So you have 10 trillion bacterial cells in you or on you every moment of your life. So the truth is you're only 10% human, you're 90% bacterial. But you guys are all crack science students, and so you know it's not really your cells, it's your DNA, right? So it's your genetic material that gives you your oomph and your personality. So what we've done is we've redrawn our man in the A's, T's, G's, and C's, right? The nucleotides that make up the human, or every genome. And so of course we have the sequence of humans, and there's 30,000 genes. We know how many genes there are in the human body. So it takes 30,000 genes to make a person. Well, if we count how many bacterial genes are in you or on you, there are 100 times more bacterial genes in you and on you, and those genes encode proteins that do things, that have functions, just like your genes do. So I don't know which of these metrics you like, but at best, you are 10% human. Most likely, you're really only 1% human. 99% of you is bacterial. It's just that we don't realize it's there because it's just a part of our day-to-day invisible life. So you have I hope you're convinced that you have all these bacteria all over. in and on you. And so the question is, what do? Well, most of these bacteria are good for us. They're living in a mutualistic symbiotic association. And because they have that vast repository of genes that I told you about, they encode all kinds of functions that we need, but we don't have the genes for. And so the one that you hear about the most is that we have these bacteria in our gut that help us to digest our food, right? They help us do things. So for example, you have Escherichia coli in your gut and it helps you digest your food, but it also makes vitamin K. So you need vitamin K to be alive. You need it for certain enzymes, and it turns out that you can't make it. But E. coli has the gene to make that, and it provides it to you. So you don't actually have to be petrified that you're covered with all these bacteria. It turns out you're supposed to be, and they provide us all of these beneficial and essential functions that we have not evolved ourselves. But of course, you have heard about bacteria. Actually, you even heard about them this morning, because sometimes bacteria get in or on you and they don't belong there. And so this happens infrequently in our lives. And when you get invaded by a bacteria that isn't one of these commensals, we call that pathogenic. And so there are many pathogenic bacteria that don't belong in or on you. And when they get in or on you, they unleash some of these factors that they have for surviving in nature, but those factors make us sick. And so I don't want to be Pollyanna about this. Most bacteria are good, but indeed we have a terrible global problem with pathogenic bacteria and infectious diseases. And so the one on the right, that's a picture of tuberculosis, the one that Dr. Tejan was telling you about this morning. So a terrible pathogen. So the question that I want to pose today, and that my lab is curious about, is whether you think about these bacteria because they're doing good things, or you think about these bacteria because they're doing bad things. The question is, how can they do anything at all? They're incredibly tiny, so how can they get any bang for their buck, since they're such small organisms? And so we want to try to understand what they're doing, what the host is doing, and how these organisms live in a community between a eukaryote and this microbial world. And so what I want to try to talk about now is how can we possibly understand this complicated association? And so here's a slide you've already seen. So you have these thousands of species of bacteria in or on you. And even though the ultimate goal is to understand what all of their jobs are and why we need those bacteria, it's hard to study that. And so what scientists try to do is we try to get our hands on model systems, like systems that approximate this complexity, but they're much simpler. And then what we hope is that if we learn about more simpler associations, step by step by step, we can learn about these more complicated situations that are sort of hard to study all at once. And so what we do is to look for what we call a model system. So what would be ideal in my mind to study these bacteria-host relationships is if we had a one-to-one relationship. So if we had a eukaryotic host, so my simple man, the stick figure, so a simple host that lived in a one-to-one association with a particular bacterium, then we could study the host and we could study the bacteria and hopefully get insight by studying that one partnership into partnerships that are much more complicated, like the microflora on our bodies. And so what's wonderful about hunting down these model systems is that nature provides these for us. So in fact, such a one-to-one relationship exists, and it's a crazy one. It's in the ocean, and it's between this animal, which is called the Hawaiian bobtail squid, and a bacterium named Vibrio fischeri. So Vibrio fischeri is a symbiont of this squid. So one animal, one species of bacteria. And what's especially good about this system is something that the bacterium does. So this bacterium is bioluminescent. It makes light, like fireflies make light. And remember, bacteria are invisible. So the things that they do are even more invisible than the bacteria. And so what's fantastic about bioluminescence is that it makes the invisible world visible. We can just see light with our eyes. We can measure it, and we can use it as a reporter that the bacteria are there and of things that they are doing. So now I want to tell you about this partnership, and what I'm going to do is first tell you about it from the squid's point of view, and then tell you about it from the bacterial point of view. So first, the squid, if we can have this movie, please. So this squid is a nocturnal animal. And so what we've done is we've put this poor guy in his water during the day, and he doesn't like it. So he looks kind of uncoordinated. He looks a little nervous because he's not supposed to be out during the day. So what he likes to do is this. He likes to bury himself in the sand and sleep all day. This also keeps him away from those nasty snails and toads. So he buries himself, and he's thorough. He wants to be buried. He wants to be hidden during the day, and he sleeps. And so he's not quite done, even though he already has camouflage. But yeah, right. So he already has camouflage, but even that's not good enough. Those snails hunt those things down. So anyway, he has to totally cover himself, and then he's going to retract those tentacles, and that should happen in a minute. And if you didn't know where that squid was, you wouldn't know where it was, right? You know where it is because we're zooming the camera out. So this is the life of the squid in the day. It just sleeps away, hidden, invisible during the day to escape from predators. The problem is you have to come out to eat. So he comes out at night to hunt. And this squid is not invisible at night. So this squid lives in just sort of shallow, knee-deep water. And so the light that the moon or the stars make, since this water is so shallow, can penetrate the depth of the water that the squid lives in. So if you were a predator under this squid and you looked up, that's what you'd see. It's a giveaway, right? And likewise, if you were just swimming, minding your own business under this squid, and the squid happened to swim over you, it would cast a shadow. That would also alert the predator that the squid is there. So this is where the bacterium comes in. What this squid has evolved is a specialized light organ that houses this Vibrio fischeri bacteria at something like 10 to the 12th cells per mil. So on the left-hand side of the picture, that's the squid sort of like you saw it. On the right-hand side, we've turned the squid over. That's the eye on the bottom. And then up on top, that big black blob, that's a specialized home for this Vibrio fischeri bacterium. So the bacterium lives in that light organ at incredibly high cell numbers. So the selection evolutionarily for the bacterium is the squid feeds it and lets it grow to numbers it could never achieve in the ocean free living. So the bacterium gets to make more sons and daughters. The squid is interested in this partnership because it wants the light from that bacterium. So the way that this symbiosis works, so now there's two pictures of the squid, the one if the bacteria wasn't there, but what the squid does is it uses that bioluminescence that the bacteria has, and it shines that bioluminescence out from its lower half. So if a predator looked up, because it's countershading itself to make it look like the water, it's much harder to find. And likewise, because it's got this beam of light coming down, what it does is it countershades its shadow. So it has two ways to keep itself invisible from the predator. Can't see it when it's up, and you can't see it when it's down. So it's an amazing anti-predation strategy. So the squid feeds the bacterium, and the bacteria like that, and then in exchange, the squid gets bioluminescence that it doesn't have the genes to make. And so now I want to go through the day-night cycle that this squid uses, because there's an interesting part about the bacterium here. So on the left, the blue line, that's the number of bacteria in this squid-like organ. So we're starting in the morning, right? The squid has buried itself in the sand, and there's very few bacteria in there. So it wouldn't matter. The squid doesn't need light. You need, so it doesn't matter. There's only a few bacteria. The squid is feeding them, so the bacteria grow and grow and grow during the day. Then at night, when the squid has to come out, there's lots of bacteria in this, 10 to the 12th per mil bacteria in this light organ. They can make a lot of light, enough light to counter-illuminate that squid. But the squid can't keep that going forever because it's too hard to feed all of them. So what happens is when the sun comes up the next morning and the squid's going to bury itself in the sand, it has a pump that's attached to its circadian rhythm. And so when the sun comes up, it pumps out 95% of the bacteria. It sort of looks like toothpaste coming out of the side of the squid. So now these bacteria are dilute, but of course the squid isn't really using them. It's asleep in the sand. It's growing them so that at night, they've grown up to high cell density. There's a lot of light there, and the squid uses it. And this happens over and over and over during the life of the squid. And it's how it keeps this bacterial culture fresh and growing and glowing when it needs it. And now I want to move to the part that is my actual favorite part, which is what are these bacteria doing? So it turns out that the bacteria are pretty smart about this relationship as well. So the answer to the question is, what is also amazing about the bacteria is that they don't make light all the time. So the observation is when the bacteria are alone, so when they're dilute, they make no light. But when they grow to a certain cell number, high cell density, all the bacteria turn on light together. And so this makes sense in the terms of that squid. Remember, they're only at high number at night when the squid is using the light. So that makes sense, again, from the squid's perspective. But the question for me in my lab is, how can it be a bacterium that is supposed to be so primitive? How can it know times when it's alone from times when it's in a community and then do something different under those two conditions? And so what we have learned about it is that the trick they use is that they make and release small molecules that we call autoinducers. And those are depicted by these red circles on the slides. You can think of these like hormones. So they make and release these molecules. And when the bacteria are alone with their low cell number, the molecules just float away. So the bacteria don't perceive them. So they don't make light. But when the bacteria grow to higher numbers, since all the members of the community are participating in making and releasing these molecules, the molecules increase in concentration externally in proportion to cell number. And when the molecules hit a certain amount, the bacteria detect that they're there and they all turn on light together. So the way that the bacteria know when they're alone from when they're in a group is by whether or not those molecules are there. They're using those molecules as a proxy for cell number. They actually have no idea how many other cells are there. They believe, if you will, that because these molecules are there, it means they have neighbors. And so we have a name for this. We call this quorum sensing. The bacteria vote with these molecules, these chemical words. They talk to each other, they vote, they put their vote out there, it gets counted, and then everybody responds to the vote in a unified group, a quorum. And so now my guys, my lab, would love to study that. We think it is amazing that bacteria have the genetic wherewithal to be able to talk to each other with a chemical language and do things in synchrony, do things as a group. And so now we want to actually have an even simpler model system. We don't want to study Vibrio fischeri that only makes light in the squid. We want to study this quorum sensing on its own. And so in order to do that, we have to go back to these evolutionary trees, this business we were talking about before. And so what we've done is we've just taken a little branch of the big bacterial tree and we'd have just some of the Vibrios. So there's lots of different Vibrio species. And so what's interesting is that all of these Vibrios, and in fact, probably all bacteria have quorum sensing, but what's important for now is that all of these Vibrios have quorum sensing. They talk to each other, but they have evolved to use quorum sensing for the different niches that they occupy. So Vibrio fischeri on the top uses quorum sensing to make light inside of that squid. On the bottom, Vibrio cholera, that's a pathogen you've probably heard about that causes the disease cholera. It uses quorum sensing to control pathogenicity. But in the middle is a bacterium that we like a lot named Vibrio harveyi. It uses quorum sensing to control bioluminescence, but it does it free living in the ocean. And so now we have a bacterium that makes light, that makes itself visible to us, that uses quorum sensing, and that does it free living. And so we want to use that bacterium now as the model to think about this in the molecular terms from the bacterium's point of view. And so we grew up some Vibrio harveyi for you this morning, and all the lights are going to go out in the room, so don't worry. And I'm going to show you this bioluminescence, I hope, that these bacteria make. So if the lights can go out. Okay. There they are. All right. Can you guys see that? Right? So those bacteria are in those flasks. They're quite dense. Those autoinducers have been released. The bacteria in there recognize that those autoinducers are there. And in response to that, they're turning on bioluminescence because they realize they're in this crowd of other bacteria, all of which is dictated by whether or not that little chemical, the autoinducer, is there. So those are the glowing bacteria and that's because of those autoinducers. Okay, so now we have this wonderful model system where we've extracted the bacteria from this host association and we can just study quorum sensing. And so quorum sensing controls bioluminescence. I think you believe that now. But what is interesting is that when a bacterium makes the switch from I'm going to go alone to I'm going to act as part of a community, it takes a lot of genes to change. It's a big deal to change from the program of gene expression that allows you to thrive when you're alone to that that lets you thrive when you're there in a community. So even though light, bioluminescence, is what we can see as the readout of quorum sensing, we've gone in and studied these more invisible traits. And it turns out in Vibrio harvii alone, over 70 genes are controlled by quorum sensing. So the genes that encode luciferase, the enzyme that makes that blue light, that's quorum sensing controlled, but also the ability of bacteria to take iron up from the environment. Bacteria have to put a molecule outside of them and get iron and bring it back in. They like to change their DNA, exchange DNA. This helps them evolve faster. And so when they give their DNA away, and then likewise, they have to secrete lots of different exoproducts that help them consume nutrients from the environment. All of those genes are controlled by quorum sensing in Vibrio harvii. What we're also learning is that this is not some weird anomaly that has to do with bioluminescent bacteria. We are now learning that quorum sensing is universal in the bacterial world. So both beneficial bacteria use quorum sensing, but so do pathogens. And so we just put one pathogen up there, Pseudomonas aeruginosa. This is the pathogen that kills people who have the disease cystic fibrosis. So what Pseudomonas does with quorum sensing is it makes a biofilm. Remember we talked about those, a biofilm on the person's lungs. So it sits down on the surface of their lungs. It secretes this goop. It's called alginate that covers the biofilm and keeps it so antibiotics can't get there. And it also secretes 50 or 100 proteins that are virulence factors that chew up the person's lung tissues. It's just trying to get nutrients. But the problem is that those toxins damage the host's lungs. So this is a terrible disease, but if you think about this from the bacterium's point of view, it is a fabulous strategy, both whether it's beneficial or pathogenic, for bacteria to be able to have an influence on their environment. All of the genes that are controlled by quorum sensing are ones that encode proteins that the bacteria have to put out there. And so you're never going to get your own back. If I give you some protein for feeding, I'm not going to get it back. But if you participate, and we all work together, what quorum sensing lets bacteria do is to be multicellular and to carry out tasks as a collective and accomplish feats that they could never accomplish if they simply acted alone as individuals. So quorum sensing is all about these kinds of traits that you need lots of cells together or the trait is completely unsuccessful. And so that's for good or for bad. And so we are interested in understanding that, like how did collective multicellular behaviors evolve on this earth? Again, we like the idea that we can study this in bacteria because they're so much simpler than real multicellular organisms. So my lab is interested in thinking about the molecular biology under that. So the question is, how do these bacteria, just like us, how do we process information from the environment? How do cells get information? And it turns out that signals come in from the environment. So things change in our environment and then of course also in the bacterial environment. And then we have mechanisms for interpreting that sensory information and behaving correctly. So in the case of bacteria, typically, if something happens in the environment, something changes. In today's example, we're talking about quorum sensing, so that would mean whether or not that autoinducer is there. But this is true for many different sensory stimuli, but just think about the autoinducer. So whether or not that molecule is there, that gets detected by a membrane-bound protein that's called a receptor. And the receptor actually goes through the bacterial membrane. So it has a portion of the protein outside and a portion of the protein in the cytoplasm. So transmembrane receptors connect the outside world of the bacteria to the inside world. And so they receive the information and they send information to the DNA to turn on and off genes, which encodes functions which allow bacteria to respond appropriately to whatever happened in the environment. And I just want to say, I'm going to tell you the mechanism for this signaling in a minute. It is exactly the same biochemistry that your cells use to send signals to tell your cells what to do and when. And so even though this evolved in bacteria, we can learn about higher organisms just by studying the biochemistry in bacteria. So now we're going to take this step by step. So the first thing is that there's this membrane-bound receptor. It's sort of the nose, sticking out there waiting for something to happen. And membrane receptors have two jobs. One is to sense a signal, a stimulus, and the other is to send that information to the DNA. And so the way that they do that is by changing their internal activity. So the receptors can be kinases or phosphatases. Kinase means put a phosphate on someone. Phosphatase means steal a phosphate from someone. So if you look at the left side, when the receptor is a kinase, it's going to take phosphate, which we have as that yellow circle, and put it on a protein, in this case, that's bound to the DNA, and then that protein can go turn on or off genes that the bacteria needs. Phosphatase, on the other side, is when the receptor takes the phosphate off of that downstream protein, and now the protein can't act. So kinase means put phosphate on, phosphatase means take it in the reverse direction. Okay, so that's how receptors work. And so now let's put this into our quorum sensing circuit. So at low cell density, so remember, this is when that chemical word, that autoinducer, isn't there. When the molecule is not there, the receptor is a kinase. It puts a phosphate, the yellow circle, on itself, but then it has to get that information into the cell. So what happens is it sends the phosphate to a connector protein called LuxU. LuxU's job is to take the phosphate from the protein that's stuck in the membrane and bring it down to a protein called LuxO, which sits on the DNA. So this is how the bacterium has connected the outside world to gene expression. It's through a kinase, a phosphorylation cascade that ultimately impinges on this protein LuxO. And when LuxO is phosphorylated, so when it has that phosphate on, its job is to shut off quorum sensing. So it shuts off light and it shuts off all the other quorum sensing genes. So at low cell density, phosphate flows to LuxO, quorum sensing is shut off, there's no light. At high cell density, so that's when these autoinducers accumulate. So remember, that's the red molecule. They bind to the receptor, and that flips a switch. It tells the receptor, stop being a kinase, be a phosphatase. And so now phosphate flow reverses. The phosphate goes from LuxO to that connector protein, LuxU, and then LuxU puts it back on the receptor. So now LuxO, the protein sitting on the DNA, is not phosphorylated, so it can't inhibit quorum sensing. So all the quorum sensing genes turn on, including the luciferase genes, which make light. Okay, so whether or not the cells are going to do quorum sensing is whether or not those molecules are there, and it just matters which way phosphate is flowing in that circuit. And so now I want to take you one more step and tell you, what does that protein LuxO do? What does it mean to inhibit quorum sensing? So the first part, the first line, you've already seen this. At low cell density, phosphate flows to LuxO, and its job is to inhibit quorum sensing. What LuxO does is to inhibit the production of a special protein called LuxR. LuxR is the master regulator of the entire quorum sensing response. So LuxR is a protein that sits on DNA and tells all these 70 genes to turn on or off. So at low cell density, LuxO stops LuxR from being made, so there's no quorum sensing. In our little example of bioluminescence, that means that the Lux, that's luciferase, the Lux Operon, so the piece of DNA that encodes the enzymes that produce light, don't get transcribed. There's no LuxAB protein, those are the enzymes that make light, and so there's no light, right? So low cell density, there's no LuxR, there's no quorum sensing, there's no light. At high cell density, remember, phosphate is going to go backwards through the circuit. When LuxO is dephosphorylated, it can't inhibit. So now what happens is that now LuxR gets made. And remember, that's the master factor. So LuxR then tells RNA polymerase, come to all these genes, transcribe them, translate them into proteins, and we change into high cell density mode. In our case, the important one is the Lux or luciferase operon. So LuxR gets that transcribed and translated into luciferase, which is called Lux A and B, and the bacteria make light. So what you're seeing first, this is just DNA in the bacterial cell, and we've color-coded the genes. So that's the Lux operon, There's 5 genes in that operon, and somewhere else on the DNA is encoded the gene for that master protein LuxR in purple. So the first thing that's going to happen is that RNA polymerase, that's RNA polymerase, the green protein, is going to come to the DNA encoding LuxR. It gets transcribed into a messenger RNA, that's the molecule you'll see now, and then ribosomes come onto that messenger RNA and they translate the message into protein, right? So now the pink protein, that's LuxR, the master transcription factor getting made in the cell. When that protein gets made, which is only at high cell density, it's going to go and tell all these genes to get turned on so the bacteria can switch into quorum sensing mode. We're showing you it getting on to the Lux operon. Its job is to tell RNA polymerase, come here, come here, come here. This is a gene that we want to make the protein now. So it's sitting on the Lux operon. Here comes RNA polymerase. And then it's going to transcribe this long operon. There's 5 genes on this messenger RNA, so they're in colors for you to see this. And then the ribosomes get on at each gene and they translate the message into proteins. We're just showing you 2 important proteins that are called Lux-A and Lux-B. Those are 2 proteins that fit like locks and keys, and when they get made at high cell density and come together, they give off photons of light. So they lock in together and light gets produced, which is what you saw in the demonstration. But of course, these bacteria are machines. It's not just one, transcript getting made. There are thousands of copies of LuxR on thousands, turning on thousands of messages to get made so that the cell can make thousands of copies of luciferase so that there's enough of those Lux-A and B proteins coming together that the entire cell actually makes perceivable light. And so that's what we're showing you there. And then if we zoom out of the cell, remember this is high cell density, so the outside world of the cell, those autoinducers are there, the bacteria know they're at high cell density, they want to switch into quorum sensing mode, enough luciferase gets made that the cells turn on light and you can see it.
So quorum sensing in bacteria involves multiple things. Bacteria can sense their density using quorum sensing factors. And these are also known as autoinducers. The autoinducer binds to receptors on bacteria that are kinases. The kinase activity of the receptors is blocked by the binding of the autoinducer. At high density, some bacteria emit light by expressing luciferase, as Bonnie has described. This operon is controlled by the translational regulation of the LuxR gene. And an antisense RNA is produced when the quorum sensing factor is low, blocking the translation of LuxR. And here I'm going to show you an animation of how the regulation of the Lux operon is accomplished by an antisense RNA.
So starting here, the cells are at low cell density, so they are making the autoinducers, but of course they live in this big world, and so the autoinducers diffuse away. So if we hone in onto the bacterial membrane, those are the receptors, and it's very infrequent that they're going to bump into one of these autoinducer molecules because those have floated off. So on the inside of the cell, remember, in the absence of autoinducer, the receptor is a kinase. So it takes a phosphate, puts it on itself, that's what it's doing now, it phosphorylates itself, and then it's going to transfer that phosphate down the line. So here comes Lux-U to take the phosphate from the receptor. Lux-U is going to put the phosphate on Lux-O, which is sitting on the DNA, waiting to get that small RNA transcribed. So there it is, that RNA gets made, and then that RNA makes a one-to-one pairing with the messenger RNA for LuxR. So whenever a LuxR message gets made, which you're seeing here, RNA polymerase make the messenger RNA for LuxR, that small RNA immediately finds it. And they make a pairing, almost like what you're looking at, and now the ribosomes can't get on. And so no protein gets made. So it's a fabulous way to stop that protein from getting made at low cell density. At high cell density, lots of cells, lots of autoinducer. So the frequency of one of those small autoinducer molecules bumping into that receptor increases enormously. So now these receptors start binding to the molecules, and then remember that event, binding the chemical, tells the receptor, don't be a kinase, switch and be a phosphatase. So now the receptor wants to slurp that phosphate out of the cascade, so there's LuxU stealing it from LuxO and putting it back on the receptor. So now LuxO is missing its phosphate, and it is dead. No short RNA gets made, so the LuxR protein can get transcribed from its gene. So here comes RNA polymerase, and it's going to transcribe the LuxR message, but that small RNA is gone. That inhibitory small RNA is gone. So now when a ribosome comes by, it's just going to get on there and translate that message into the protein LuxR, which was the important protein we talked about yesterday that runs the show. So now LuxR goes to all the DNA encoding target genes for quorum sensing. In this case, of course, we're showing you my favorite ones. These are the genes for luciferase. It makes messages for all of the proteins the bacteria needs for quorum sensing. In this case, this is the Lux operon. And there's two important proteins we showed yesterday. Those are called Lux A and Lux B, right? So those are getting made. And then, of course, those two proteins are going to come together, and when they make this touch each other and make this hand-to-hand configuration, the bacterium makes light. And so we learned all this in Vibrio harvii, but the cascades are similar in pathogenic bacteria. You just have to substitute nasty outputs for that beautiful bioluminescence.
Here are some genetic terms and concepts I think you should know. Gene regulation. Mechanisms and processes that control the phenotypic expression of genes. Regulatory gene. DNA sequence that encodes a protein or RNA molecule that interacts with DNA sequences and affects their transcription or translation, or both. Constitutive gene. A gene that is not regulated and is expressed continually. Domain, a functional part of a protein. Operon, set of genes in a bacterial cell along with a common promoter and other sequences, such as an operator, that control the transcription of the genes. Regulator gene, gene associated with an operon in bacterial cells that encodes a protein or RNA molecule that functions in controlling the transcription of one or more structural genes. Operator, DNA sequence in the operon of a bacterial cell. A regular protein binds to the operator and affects the rate of transcription of the structural genes. Inducible operon. Operon or other system of gene regulation in which transcription is normally off. Something must take place for a transcription to be induced or turned on. Repressible operon. Operon or other system of gene regulation in which transcription is normally on. Something must take place for a transcription to be repressed or turned off. Inducer. Substance that stimulates transcription in an inducible system of gene regulation. Usually a small molecule that binds to a repressor protein and alters that repressor so that it can no longer bind to DNA and inhibit transcription. Partial diploid. Bacterial cell that possesses two copies of genes, including one copy on the bacterial chromosome and one on an extra piece of DNA, usually a plasmid. It's also called a merozygote. Attenuation. A type of gene regulation in some bacterial operons. And this is, we use TRIP as an example in today's lecture, in which transcription is initiated but terminates prematurely before the transcription of the structural genes. Antisense RNA. small RNA molecule that base pairs with a complementary DNA or RNA sequence and affects its functioning. Quorum sensing is a regulation of gene expression in response to fluctuation in cell population density. Here's an image of a histone octamer complexed with DNA. And histones are very important in gene regulation in eukaryotes, and that's what we'll be discussing in the next lecture. See you next time.